Okay, so today we got the muscular system. Today was supposed to be nervous system, but then I had an hour and I slept in, and and apparently nervous system was supposed to be 1 hour, but then no, it was 100 minutes. This one was like 48, so I was like, okay, we're gonna just do muscular system instead of nervous system. So what are the three main types of muscle tissue? There's smooth muscle, there's skeletal muscle, and then there's a cardiac muscle. So, talking about the smooth muscle specifically, what is its appearance? It's striated because it has sarcomeres. What is it used for? It's used for voluntary movement. Nucleus count, these are multinucleated. What process do they undergo? This is like myogenesis. Wow, I actually don't know how I remember that. I just remember that weird detail from the video actually. Myogenesis, is that correct? Yep. What are satellite cells? Those are cells that just help repair damaged muscle cells. Like they're just like kind of like goblet cells. I would feel like related to the neurons, but I'm not too sure. Support and repair muscle cells, skeletal muscle cells. What are the three levels? Okay, this is what I'm kind of cooked on. So this is the endomycelium, endomycelium or mycelium, Okay, so the outermost part is the epimycelium. That's like surrounding the muscular tissue. Now we're gonna go inside and we're gonna have the epimycelium. No, we're not gonna have the epimycelium. We're gonna have, what's the epimycelium, bro? There is no such thing as the epimycelium. Okay. Wait, no, epimycelium is the outer one. What the heck am I saying? Okay, so we got the epimycelium that's on the outside of the muscle tissue of the entire thing. Now we're gonna go inside and then we're gonna see the myosium. Nope. Perimycelium. Perimycelium is on the inside. And the perimycelium is going to be what kind of coats around these things called fossicles? Nope. F, it starts with the F. Bro, I just can't think of the word. Fascicles. Bro, they really gotta come up with some new names. Isn't the fossicles like the ear muscle, ear bones? Then the fascicles are what are small bundles of muscle. So then within those fascicles, you're gonna have like muscle cells, and those muscle cells are gonna be covered by the endomycelium, which is surrounding each individual muscle fiber, like the muscle cell. And then within that, you're gonna have the sarcomeres, which are the most basic unit you can have. But those sarcomeres will make up myofibrils. So muscle fibers contain myofibrils, which are made up of repeating units of sarcomeres. And sarcomeres are the smallest functional unit of muscle contraction. Golly, that was rough. Okay, so muscle fiber anatomy. So we're gonna go through the muscle fiber, the muscle cell. So first we have the myofibril, then we have the sarcomere, then we have the sarcoplasmic reticulum, then we have the mitochondria, then we have the T tubules, or I feel like T tubules is not the right word. It's I think it's T tubules. Yep, T tubules, and then you're gonna have the sarcoplasm. No. Wait, no, the first S and the second S is not that. You're gonna have M sarcomere. Okay, wait, sarcoplasmic reticulum. Okay, those are right. Then you're gonna have mitochondria, T tubules. Then you're gonna have the sarcoplasm. Then the sarcoplasm is gonna go down into the sarcolemma. Yeah, okay. So what is the functional unit? The sarcomere. What stores calcium? The sarcoplasmic reticulum. What are T tubules used for? Those are used for kind of like they kind of just like expand the depolarization. Like they spread the the like the charge around. Like I'm saying, like they just transmit. They are made up of the sarcolemma membrane, transmit electrical signals inside the fibers. Okay. So what are sarcomeres made of? What are they also called? Sarcomeres are made up of actin and myosin. say a word. Actin and myosin, they are also called thin and thick filaments. Thin for actin, thick for myosin. What are sarcomeres made of? What are they also called? I already said that. Anatomy of the sarcomere. So we have the Z line, we have the M line, the I band, the H zone, and the A band. So what is all these? Well, I could tell you that the M line is the middle line. That's the very middle. The I band is where it's actin only. The H zone is the H zone. H zone is myosin only. Then the A band is the overlap between myosin and actin. Nope, that's wrong. The A band is the length of the myosin filament. That's going to stay constant. The H zone is the length of the myosin, and that is going to decrease as the contraction. And then the I band is the area of actin only, which will also shorten. The M line is the middle of the myosin filament. The Z line is what anchors the actin, and that's kind of like the definite length of the sarcomere. So let me try that again. The Z line is the segment of sarcomere. That's just from one end to the other end. The M line is the middle of the filament, the middle of the sarcomere. The I band is actin only. The H zone is myosin only, both of which shorten during contraction. The A band is the definite length of the myosin filament, and that does not change. How does the skeletal muscle grow? What is this process? What does it not go through? It does not go through mitosis. It grows through hypertrophy. What things will increase? The number of nuclei will increase, the number of mitochondria will increase, the cell diameter will increase, things and such like that. Muscle fiber size, mitochondria, glycogen, myofibrils. Well, I guess I said some stuff. Myofibrils will increase, glycogen will increase, mitochondria fiber size. Let me just put a star on that. What else does the muscle help with? It helps with moving blood. Those contractions will squeeze on the veins and stuff like that, and they'll just move the blood. So 3 types of contractions grouping for 2. So we got isotonic and we got isometric. Isotonic is the eccentric and the decentric, while isometric is just like holding it in place. So it's a great naming system. 3 types of skeletal muscle. So we got type 1, we got type 2A and type 2B. So type 1 is what you would call the fast twitch muscles. They are very red. They have they got like a high abundance of oxygen because they go through a lot of aerobic respiration. They pump through oxygen quite quickly. Those are like for long distance, like long endurance, and they're just like there, like that's just like how they are. Let me just read the notes one more time to make sure. But my computer is geeked. Where is this, bro? Ah, yes. Nope. Let me touch you tonight. Type 1 is high endurance. So these are going to be their slow twitch muscle fibers. These have lots of myoglobin and mitochondria. They go through aerobic metabolism. They look red and they have a smaller diameter. Okay. Oh, wait. I literally have it right here. What to classify them. So we have type 2A, which is going to have an intermediate amount of myoglobin and mitochondria, intermediate pinkish color. They're gonna do some aerobic and some anaerobic, and they're gonna have an intermediate diameter. So there's just the middle ground. Then we're gonna have the type 2B. This is pure fast twitch. So they're gonna have low myoglobin, like low mitochondria, They're going to be whitish in color. They're going to be doing mainly anaerobic respiration because they're going to be short bursts. So these are going to be called fast twitch muscles for high power, and their size and diameter is much bigger. So steps to contraction. What is the NT? So you're going to have a neurotransmitter called acetylcholine. Acetylcholine is responsible for the action potential, and that'll cause the depolarization of the muscle fiber. The muscle fiber will then expand. The muscle fiber will, okay, wait, where's that good thing? Okay, so I was saying acetylcholine will go to the synapse, which is called the end plate on the junction. So it's like you know the freaking junction, like the neurotransmitter, the motor neuron will attach to the end plate. This is what it's called, I believe. The motor end plate, and it'll give acetylcholine. It'll give acetylcholine to it. That acetylcholine will cause the surface to depolarize, and then that action potential will spread along the muscle. Then it'll get to the T tubules, which will take the action potential into the cell, which will cause depolarization. So Na plus will be going into the cell, right? Like, you know what I'm saying? Like, it'll like become less. Wait, let me just, okay, break down steps of contraction to know. Bro, there's so much information to know about this stuff, bro. It's genuinely crazy, bro. So nerve signal arrive, acetylcholine will release. Bro, can I scroll up? Can you stop being so finicky? Depolarization binding causes the sarcolemma. So it'll cause the sarcolemma to depolarize, triggering an electrical signal that travels into the muscle fiber through the T tubules. Yeah, so the T tubule is going to trigger that electrical signal into the muscle fiber. Now, the T tubules will cause the sarcoplasmic reticulum to release calcium 2 plus. That calcium 2 plus is going to bind to myotrypnin. No, not myotrip. It'll bind to troponin. Wow, I'm just making up names, bro. It'll bind to troponin, which is connected to myotroponin. Myosin, troponin. Wait, no, I got that backwards. Tropomyosin. Whoa, okay, let me just restart. So we're gonna, acetylcholine is gonna go, I'm trying to visualize this through my brain, so I'm closing my eyes. So motor neuron is going to take acetylcholine, put it to the motor cell, through motor neuron to the end plate. Then the end plate is going to cause the muscle, the endomysium. Okay, it's going to bind to, wait, no, myosine, that's the outside, bro. What am I even talking about? It's going to bind to a sarcolemma. Wait, what's the sarcolemma again? Bro, oh my god. Where are the fascicles? Sarcomere. The sarcolemma. What's the sarcomere? Oh, the sarcomere is this functional unit. Right? So what's the sarcolemma? The sarcolemma is what's outside. Bro, this is just genuinely... What the heck is this, bro? I'm geeked, bro. Bro, I need to just go to bed, bro. Okay, let me fucking see this, bro. So muscle contraction. Acetylcholine is going to go to the end plate through the motor neuron. That'll cause the sarcolemma to depolarize, and that'll cause the Na plus freaking things to open and everything. Then the T tubules will transmit that electrical impulse into the cell, and that'll cause calcium 2 plus to be released from the endoplasmic reticulum. Well, the sarcoplasmic reticulum. The sarcoplasmic reticulum is what's used to store calcium 2 plus. Calcium 2 plus will then go and bind to troponin, which is connected to tropomyosin, which blocks the myosin binding sites on the actin. So then once that myotroponin moves, then the binding sites for myosin and actin are open. Okay, so now the myosin and actin are going to connect. How do these connect? Well, the myosin head has ADP attached to it. That ADP will allow it to attach